Yeah. So, hello everyone. Good evening, and today we will be looking at the week 11 of the Interactomics course. Where mostly we'll be focusing on different strategies like the mass spectrometry based technology strategy, which we have already discussed in quite detail. But overall, we will be looking at how we can take it forward for our understanding or how to do some kind of analysis of what the concepts we have discussed. And we had also discussed yesterday about how the difference between the proteomics and the genomics things, where proteomics is that we need to take the fragments and then try to match the theoretical spectra with the experimental spectra and then try to find out the matches and the results. Whereas in the NGS, different technologies, different proton transfer, different page change, different signals, and starting from Sanger sequencing or the chemical sequencing method, we have identified the things. So today also we will be looking into the different repositories of which we have given in the extra session. We have discussed about one of the major thing that was the CBio portal for the cancer genomic studies, which is of very quite relevant to understand the different nutritional status and the nutritional landscapes of different genes and the proteins. But today also we will be looking into the different repositories of the human proteins, like the Human Protein Atlas, which is made, and how one can get knowledge. So actually there was in the first week, if you just recall, if you all just recall, that we had discussed that how different omics technologies were uh, made and how the technology evolved, quite starting from genomics, from the human draft map of the human uh, genome map to getting at a very high level of what we call is that taking the mass spectrometry based technologies, but then we had the tissue based draft map of the human proteome to the tissue based draft of the human proteome, or we had the antibody based draft map of the human proteome, which Human Protein Atlas played a huge role. So we will be looking and discussing about that, and also like the CBio portal, which is being used heavily for this kind of analysis. So what we generally tend to focus on, or which generally tend to will look to keep on, is that majorly not only the different technologies but different repositories are also helping us to uh, to get this kind of data, and also this helps us, the other researchers, the global community, to help a lot. So similarly, there has been also one repository. Uh, so we will be talking about that as well. So before that, moving on. So today we will be looking first on the proteomics data analysis, primary data analysis hands-on. So for which I'll be sharing one file which you need to upload and you need to start. So this concept we have read. I will just reiterate the concept and try to see that how we will be using those in our uh, data analysis or primary analysis for identification of the proteins. So what we are doing, we have protein, all proteins extracted from biospecimen, be it tissue, be it cell acid, be it serum, be it anything. So we have the protein. What we are doing, we are so first from the buffer we have extracted once we have done, we are doing uh, reduction using TCP, right? So it is getting reduced through TCP. Then to keep this reduction and not it defaults back its alkylation using IA, right? And then so this give a modification of carbamidomethylation, right? So this gives a, so why I am telling this, when we will be doing the analysis now, the primary analysis, you will be seeing, right, that how it is getting changed and what is the uh, what are the parameters that we are giving is mostly focused on this type of thing. That is, it is very much focused on the way samples are prepared and sample is getting prepared, right? So this is how we get to know the reduction and the alkylation. Then followed by what we are doing once we have alkylated the things, the carbamidomethylation is done. We are reducing it to trypsin, and once it is getting chopped down, then this is resulting into the bottom of proteomics. So these fragments of the peptides or proteins, bottom of proteomics, where these fragments are run into MS, and we try to identify their spectra. So we know that, you know, we have discussed in great detail about the tandem mass spectrometry. Either you can use some labeling technologies for quantification or not, but yeah. So the tandem mass spectrometry, which helps you to do the MSMS to get the deduce the sequence of the peptides, right? So once you have done that and want to have deduced the sequence of the peptides, so then what you want to do, you will be matching the theoretical spectra and the experimental spectra to identify the peptides and the proteins. So this is the whole concept of proteomics, but how after the sample has been run, how we are trying to identify the proteins and understand which proteins are there, that why we will be looking into the thing, right? So now we would be seeing how we can do. So for that, we would be taking one of the so we would be looking at one of the free source website, right? So we are getting the right. So I am hopeful that everyone has got this page, right? So what we will be doing, we will be doing for mascot database search. So So we are getting this access mask. So there are, you can see there are four options: access master server, peptide mask fingerprint, sequence query, MSMS and search. Right? Is there any question? No, right? So what we will be, we can go for the peptide mask fingerprint. So what is peptide mask fingerprint now? So peptide mask fingerprint is that. So if you do at a MS level, you will get only the embodied value of the whole peptide. So the peptide can be of 10 amino acid long or 15 amino acid long or 20 amino acid long. So what you are getting is that the peptide sequence that automatically you are reducing that whole length of the peptide sequence. And once you get the whole length of the peptide sequence, and definitely what you will be doing is that you will be getting the MBZ values. And once you get the MBZ values, then definitely you can say that this peptide is having this much mass, but you cannot deduce for the sequence of the peptide exactly, because many amino acid combinations can also give you the same MBZs, right? So what we are doing is that we will go with the MSMS answers because we are doing the tandem mass spectrometry, right? So perform, okay. So such title I am giving Interactomics, right? So now here you can see that there are different databases. One is the contaminants. So anyone would like to say what is the contaminants database? So contaminants database is something that which so let us first search what it says. Okay, so what is a contaminants database? First, let us look this thing here. Okay, so there are if you search a single organic database, it is usually a good idea to include sequences for contaminants such as keratin, BSA, and trypsin. Okay, just look and just read what I'm saying. If you search as a two groups make their collections available, maintains approximately 250 proteins selected from various sources. Global Proteomics Organization Common Repository of Adventure contains more than 112 proteins selected from Swiss spot, and the numbers as of October 2011. In Mascot 2.3 and later, you can simply select the contaminants database in the search form along with the target database. You need to append the contaminant target. This can be complicated by the requirement to have uniform syntax. So this is what this is. So basically, the common proteins that can come from your nails, come from your bad lab practices, different common environment. So these proteins, if they come and if they make for it, then it is a troubleshoot and a problem. So that's what we are trying to add in the contaminants database so that you can remove those kind of peptides and proteins from your data. And then we can give any of those thing. We will go with the Swiss plot. Okay. So if we don't know what is exactly the organism is of which protein we are looking at, we will go with all entries. Enzyme trypsin, quantitation none, cross-linking none. So now we had already disclosed that we will be using carbamidomethyl C as a fixed modifications, right? And now we will be choosing the data file. And we are keeping the stat. So it is something coming as the scores like the tubulin alpha 3 of Arabidopsis thaliana. So these are the peptides. So these are the fragments, Y ions and the B ions. Can you visualize it, right? The D, V, N, A, B, G, T, I, K. So these many Y ions and these many B ions have been identified. So B1 ions are very difficult to get, but B2 ions one can easily get. So this is having a protein sequence coverage of 13%. That is, this peptide has been identified, this peptide has been identified, this peptide has been identified, this peptide has been identified. And these are the peptides that have been identified, and the protein sequence coverage is only 13%, right? Let us now play with it. What we will do is that we will be using phospho ST and phospho Y as variable modifications. What is that? You know that these are phosphorylation. Though we don't know it is in enriched data or not. Again, phosphorylation gets enriched with columns like Ti2, titanium dioxide. It is coming as peak trypsin. So we had given only the contaminant database, and that's why it took the big trips in. But when we are adding both Sysprot and this thing, we will see that there is a change. So in the contaminants, there's a trypsin peak. Trypsin is also there added, trypsin sequence and all those keratin and BSA sequences. So since it is not a enriched data or phosphorylation, or we don't know, so still the coverage is doesn't increasing. So we are getting what was expected, that it is of 13% coverage Arabidopsis thaliana with this manufacturers of this particular protein, and these are the fragments that is getting identified, the Y ions and the E ions. Do you all have any question here? Sorry, so there was some technical issue. So what we are saying is that Yeah, so what we were talking about was that identification of certain proteins and the peptides. If we do that and if we download it, but uh, but the problem is that we can identify those things and we can also play with the parameters with, of course, knowing the history going back to that how the sample preparation has been done and those kind of analysis, right? So this is what we can get and we can understand and we can identify, right? So this is how to take it uh, forward. Okay, so now what we will be looking into the some of the repositories. Okay, so and how those can be utilized for different uh, important biological information. So we would be looking into that aspect. So human protein atlas to begin with. So we would be happy to know that here this India also participated in giving the pathology atlas. So we would be looking into that in detail. Just a minute. Yeah, so we'll be starting with the human protein atlas. Right. So here you can see tissue, brain, single cell pathology disease, and you can search. So you can see that what is their expression level at the tissue, brain pathology. So let us. Hmm. So tissue-based map of the human proteome. So molecular details in the different tissues will greatly increase our knowledge of human biology and disease. Here we present a map of the human tissue proteome based on an integrated omics approach that involves quantitative transcriptomics at the tissue and organ level, quantitative transcriptomics at the tissue and organ level, combined with tissue microarray based immunohistochemistry to achieve spatial localization of proteins down to single cell level. Detected more than 90% of the putative protein coding genes. We use this approach to explore the human secretome, the membrane proteome, the druggable proteome, the cancer proteome, and the metabolic functions in 32 different tissues and organs. Integrative web-based database that allows exploration of individual proteins, global expression patterns in all major tissues and organs in the human body. Okay, so the tissue-based map of the human proteome. So protein expression across human tissues. So sequencing the human genome gave new insights into human biology and disease. However, the ultimate goal is to understand the dynamic expression of each of the approximately 20,000 protein coding genes and the function of each protein. Now present a map of protein expression across 32 human tissues, and they not only measured expression at an RNA level but also used antibody profiling to precisely localize the corresponding proteins. So resolving the molecular details of proteome variation in the different tissues and organs of the human body would greatly increase our knowledge of human biology and disease. Here we present a map of the human tissue proteome based on quantitative transcriptomics on a tissue and organ level combined with protein profiling using microarray-based immunohistochemistry to achieve spatial localization of proteins. We provide a global analysis of the secreted and membrane proteins, analysis of the expression profiles for all proteins targeted by pharmaceutical drugs and proteins implicated in cancer. So we have used an integrative study, the special immune proteins, all major tissues and organs, n equal to 44, 24,028 antibodies. So this is the antibody-based strategy that have been used, corresponding to 16,975 protein encoding genes, complemented with RNA sequencing data for 32 of the tissues. The antibodies have been used to produce more than 13 million tissue-based immunohistochemistry images, each annotated by pathologists for all the sampled tissues to facilitate integration with other biological resources or later available for download and cross-referencing. So we report a genome-wide analysis of the tissue specificity of RNA and protein expression, covering more than 90% of the putative protein coding genes, complementing with analysis of various subproteins which has predicted secreted proteins and membrane-bound proteins. Analysis shows that almost half of the genes are expressed in all analyzed tissues, which suggests that the gene products are needed in all cells to maintain. This analysis shows that almost half of the genes are expressed in all the cells to maintain housekeeping functions, such as cell growth, energy generation, basic metabolic. Further, there is enrichment in metabolism among these genes. 60% of all the metabolic enzymes are expressed in all analyzed tissues. The largest number of tissue genes is found in the testes, followed by the brain and the liver. Analysis of the 618 proteins targeted by clinically approved drugs. 30% are expressed in all analyzed tissues. 30% are all expressed in all the analyzed tissues. An analysis of metabolic activity based on genome scale metabolic models revealed liver as the most metabolically active tissue, followed by adipose tissue and the skeletal muscle. So among the 618 proteins, 30% are expressed in all present tissues. So 30% of 618 is so around 185 proteins are expressed in all. So there will be off-target effects and those things, right? So this is a freely available interactive resource is presented as part of the Human Protein Atlas, offering the possibility to explore the tissue-elevated proteomes, tissue-specific protein class, comprehensive expressed at elevated levels to provide a special context with localization of the proteins at a single cell level. So resolving the molecular details of proteome variation in the different tissue and organs of the human body will greatly increase our knowledge of human biology and disease. Human tissue proteome based on an integrated omics approach that involves quantitative transcriptomics at the tissue and organ level combined with tissue microarray based immunohistochemistry to achieve spatial localization of proteins down to the single cell level. Our tissue-based analysis detected more than 90% of the putative protein coding genes. We use this approach to explore the human secretome, the membrane proteome, the druggable proteome, the cancer proteome, and the metabolic functions in 32 different tissues and organs. All the data integrated in an interactive web-based database that allows exploration of individual proteins, global expression patterns in all major tissue and organs of the human body. So there is much interest in annotating all human genes at the level of DNA, RNA, and protein with the ultimate goal of defining structure, function, localization, expression, and interactions of all proteins. Large-scale projects such as ENCODE and the Human Protein Project aim to integrate results from many research groups and technical platforms to reach a detailed understanding of each of the 20,000 human protein coding genes, human genome, and the corresponding protein forms. Recently, drafts of the human proteome based on the proteogenomics efforts have been described. A human protein atlas program which is exploring the human proteome using gene-centric and genome-wide antibody-based profiling on tissue microarrays. This allows for special pathology uh, Yeah, so if we look into it, so Human Protein Atlas exploring gene-centric classification of protein coding genes. So we can see analyzed in study including tissues, studied both by RNA-seq and antibody-based profiling, and those analyzed not only by antibody-based profiling for heat map showing the pairwise correlation based on transcript expression levels for coded of individual samples and number of genes in each expression category according to the Venn diagram, the distribution of genes. The transcriptomics refine the classification performed earlier. Tissue elevated proteome, human secretome, and the membrane proteome. Right? So we can go through it now. We'll say the human protein atlas is a tool for pathology, right? Tissue-based diagnostics and the research is incessantly, which is your increasing with the development of new molecular tools. Long as that immunohistochemistry can add an important new level of information on top of morphology. Availed crucial diagnostic and prognostic information. So cell lines altogether 708 spots. Human protein atlas, human proteome pathology, transcriptomics to ambitious proteomics efforts, resulting in rapidly growing knowledge base has created a demand to transform omics data into biomedical research and the increase of information. Together with the advancement of technologies, reached a point where functional studies in model systems with the analysis of human tissues, antibody-based proteomics, immunohistochemistry, and the tissue microarrays. Invaluable method tools for in situ visualization of protein expression patterns in tissues and cells. Protein profiling, human protein atlas portal, biomarkers, and those proteins. Yeah, so now just moving on. So we have, so you can go through human protein atlas, which is of very useful and fruitful for developing those kind of things. So we have now, if we look for the analysis of the proteomics, once we identified the protein, so now to have a better understanding of the statistical power test. So it is the probability of correctly rejecting the null hypothesis when it is called. Measures the abilities to detect a true effect or difference when it exists. Suppose we want to compare the mean scores of the two groups, group A and group B. We could conduct a t-test to determine if the mean score of group A is significantly different from the mean score of group B. The null hypothesis for this test is that the means are equal. Significance when we 5% chance. So suppose we have a sample of 50 individuals, mean score 75, mean score is 80. For both the groups is 10. Using a two sample t-test, Steve p-value of 0.028. To determine the statistical, we need to specify the effect size and sample. So we want to achieve a power of 80.8% with an effect size of medium effect size. We want to achieve this level of power. So you can do that here. Yeah, so you can see that you can calculate and we can